Hello and welcome to our iGEM Science Slam 2020. My name is Angela and I will guide you through this evening. However, it's not about me today, but about the research of our presenting teams. This is why I want to welcome them now. Today we have guests from the University of Lausanne in Switzerland, from the University of Manchester, the Imperial College in London, and the Ashesi University in Ghana. We are extremely happy to have gathered such a group of young and enthusiastic people from all over the world presenting their research to us today. So thank you very much for being here now. Talking about teams, I guess there's still a question mark on the face of many of you spectators. iGEM is a competition in the field of synthetic biology. For the moment, I want to leave it at that, but later we will come to this back again, and then I will explain what iGEM is in more detail. But for now, let's move on. So, how does our program look like today? Let us go through this relatively quick. For those of you who have never seen a Science Slam before and don't know how it works, I briefly explain the rules. Today we have 5 speakers. All of them are iGEM team members presenting their project or parts of it in an exciting manner. Each slam will take 10 minutes. After 9 minutes, we will give a short signal so that you know you're on the final sprint. In the end, we will have one winner team, which you can choose at home. How this will work, I explain this later when we arrive at this point. In the end, we as the host will also present our research. But of course, we won't compete for the trophy. One last thing I want to say before we finally start. We are a little bit sad that we don't have you in front of us and we don't see your faces and we don't see your reactions. So please, if you have any questions, any feedback, if you want to tell us anything, don't hesitate to write it in the comments. We are really happy to read them and we really will read them. So do that, please. And now I say we start. Our very first team is the iGEM team from the University of Lausanne. The speaker is Ilinka, and what her team is researching on is a special kind of cancer therapy. I don't want to take anything away from you in advance, so this is why I want to say, Ilinka, the stage is yours.
Great, thank you. Okay, so hi, I'm Alinka, and today I'm going to be presenting bots. I'm the student leader of the iGEM team of the University of Lausanne. This is our first time competing, so it's been a very intense year to start, but that's not what this is about. So, what is bot? Okay, there we go. Sorry. So BOT stands for Bacterial Oscillation Therapy, and the idea behind this is to use an engineered bacteria that can produce a drug in a repetitive way to treat colorectal cancer. So how is this supposed to work? The general idea behind our project is that our bacteria can be ingested by the patient and it will travel safely down the digestive system and colonize tumors in the colon. The great thing about this is that the bacteria that we're using actually already colonizes tumor-like environments very easily and very efficiently. Once we get into the colon, it's hopefully going to be something like this, with our bacteria in blue, and it produces our drug, azurin, which inhibits tumor growth. So this is it's a lot to take in. Like I said earlier, we wanted to produce everything in an oscillatory manner, and this is what we can see in the bacteria. It's basically just the way that we are making our bacteria produce azurin every 24 hours roughly. The reasoning behind this, I'll go into detail a bit later, is there's many studies that have shown actually that this can have very high effects on the efficiency of the drugs. So we are using azurin, which actually comes from a very different bacteria and can basically stop tumor growth. But to talk about that, we have to talk about what is cancer. So cancer arises from a dysregulation of the cell, usually in the cell life cycle, and this leads to uncontrollable growth, and it makes the cells gain immortality. So basically they don't die in the same way that they should whenever something goes wrong. Cancer has many ways of developing. The most common is through changes in the DNA of the cell. These are known as mutations, and these can occur through specific exposure to certain situations or just through cell life. Why did we pick colorectal cancer? Because over 1 million people are diagnosed every year with this disease, and it accounts for about 10% of deaths, cancer-related deaths in the same period. The thing with colorectal cancer is because it's in the colon, it can affect multiple processes such as absorption of nutrients and water, excretion of nutrients, and also just general metabolic processes. And this can lead to having just even more problems on top of just having cancer. So this is what we want to treat. So how do we want to treat it? So as I mentioned earlier, we want to use azurin, which is a bacterial protein. The way that azurin works is that it is very specific to cancer cells, and it gets in very, very easily. Once it gets into the cancer cell, it binds to this thing called p53, another protein. It's very famous. Can't really learn anything about cancer biology without learning about p53. The way that p53 works is that it accumulates in the cell when something goes wrong, like if DNA is damaged. And once it accumulates, it can actually lead to cell death. Now, cancer cells have a way around this. But azurin binds to p53, stabilizes it, allows it to accumulate, and this leads to cell death, or as we like to call it, apoptosis. So if azurin works so well and is so specific, why would we want to have a timed delivery? The reason for this is that studies have shown, as I mentioned earlier, that timing the delivery of your drug can actually increase its toxicity to the tumor cells whilst lowering the side effects and lowering the effect on the healthy surrounding tissue. And finally, why did we decide to use a bacteria for this? Our bacteria is very specific. As I mentioned earlier, it colonizes tumors very easily. And because the idea is to take a pill, it means that it becomes very much less invasive for the patient. So in conclusion, with our iGEM project, we hope to combine multiple domains towards a common goal, which is treating cancer. But of course, this project would not be possible without the help of our sponsors, which we would also like to thank.
So thank you very much for that presentation. I think now everyone has a better impression what an iGEM team is doing. Therefore, we should talk a little bit more about iGEM. iGEM is a huge international competition in the field of synthetic biology. This year, we have about 257 participating teams and about 5,000 students behind those teams. So These teams are working for months on problems in the field of synthetic biology. Thereby, we have a wet lab team who is working on planning the experiments and conducting the experiments. And we have a dry lab team who is doing things like mathematical modeling and machine learning and stuff like that. What we are all looking forward to is the giant jamboree in November. This is the final event for which teams travel from all over the world to Boston to present their research to several judges. However, iGEM is not only about winning a medal, but also about the community. So during the Giant Jamboree, there are also many workshops and possibilities for the participants to exchange their experiences, to talk with each other, and to learn something from each other. However, as you can imagine, this year, due to Corona, everything is a little bit different than usually. So we had a couple of challenges to face, and we still have. For example, teams were struggling to get lab access, or team meetings are held online. By now, many teams already managed to start experimenting in the lab, but there are still projects who are dry lab only. Moreover, we are a little bit sad that we won't travel to Boston, but we have a virtual Giant Jamboree to which we are also looking forward to. So let's come back to the Science Slam. And now I want you to think of a really hot summer day, maybe at the beach, maybe you have a little bit of ice cream in your hand, okay? So in summer, it is very popular to go out and get a tan. I also like to do that. And what I find interesting is that people deal with the problem of getting a sunburn really differently. So when I look around at the beach, I see people who are already white from all the sunscreen and who always use the highest protection factor. But what I also see are people who don't really care about getting a sunburn and who don't use sunscreen at all. However, the focus is always on the danger of the sun for the human being. But people never talk about the danger of sunscreen to other human beings or living beings. However, the iGEM team of Manchester does. And in the next presentation, they will talk about why and how sunscreen can be dangerous for living beings. So stay curious, and now the stage is yours.
So, hi everyone, my name is Miguel, and I'm going to be presenting this with my teammate Mari. Firstly, thank you to Heidelberg for putting all this together and inviting us to join. We are going to be taking you through some of the major aspects of our iGEM project, Hyposol, the sunscreen for the future. So first, I'm going to take you through the structure of our project. Basically, the sun emits UV rays that pass through the Earth's atmosphere and are absorbed into our skin cells. UV radiation results in DNA damage, gene mutations, suppression of the immune system, and inflammation. All of which have important roles in photoaging and on the onset of skin cancer. For example, UV radiation mutates the p53 tumor suppressor gene, which controls the death of potentially cancerous cells. We can protect our skin with sunscreen, which contains UV filters in its formulation. These filters are released into marine ecosystems, where they cause an increase in coral bleaching. The African hippopotamus produces a natural SPF compound called hyposudoric acid, which acts as a UV filter in the hippo's sweat. This prevents these huge hairless animals from burning while relaxing in waterways. We plan to mass produce hyposudoric acid using E. coli bacteria to use as an alternative UV filter in a reef-friendly sunscreen.
So conventional sunscreen compounds, such as oxybenzone, enhance the viral activity in the seawater by inducing the lytic cycle. The increased viral activity results in stress and the coral bleaches. The identification of oxybenzone and other sunscreen components in waters cause concerns not just for coral but for many other aquatic species because they're not removed via standard wastewater treatments. This results in the bioaccumulation of oxybenzone and other toxic compounds found in sunscreen along the food web. If this is allowed to continue, it could result in the gradual destruction of the marine food web and issues for human health. Therefore, our solution is to minimize the oxidative stress caused experience by replacing conventional UV filters with our own novel filter, hypohydronic acid. This year, we were lucky to be recognized by Revive and Restore, who are dedicated to survival of species through genetic rescue. Our project's ultimate goal is to reduce bleaching events because this shallow water ecosystems is a dependence on the symbiosis between algae and corals. Corals that are not killed in bleaching events have reduced growth and calcification repair capabilities. These effects ripple out into the reef, reducing the overall reef productivity. This leads to other marine organisms that depend on the reef to survive. These ecological effects have not been properly assessed and could potentially be detrimental to the entire marine ecosystem. It is our goal that when using our filter, the oxidative stress on corals reduces, meaning less algae is expelled. This increases the fitness of corals, helping them to better support the organisms of that ecosystem. Overall, it has positive effects on fitness, productivity, and biodiversity. This slide here shows an example of a completely bleached reef. Because the algae has been expelled, the coral loses its color and dies.
So I'm going to talk a bit about our compound here. Hypohydronic acid is a broad spectrum that goes from 280 to 400 nanometers UV filter, meaning it protects both against UVA and UVB radiation, and it fits current sunscreen legal protection requirements. We are making assumptions that hypohydronic acid is reef safe due to its presence in the marine environment. However, runoff from rivers into the sea would mean hypohydronic acid is present in very small concentrations. Part of our plan next year, when we are allowed in the labs, is to test the effect of hypohydronic acid on coral at increasing concentrations. We also assume that it is consumer safe, as it is mammal-derived compound that acts on the hippo's skin. We have also heard of someone drinking hippo milk, which also contains high levels of hypohydronic acid, but we cannot find that video anywhere. Testing this compound on human skin would be part of the final stages of our project next year. Finally, hypohydronic acid is believed to have some antimicrobial effects. This will help to reduce infections when children cut themselves on rocks at the beach, giving our sunscreen extra benefits. Hypohydronic acid is a very new compound that has not been mass-produced in bacteria before, and the pathway used by the hippo for its production is uncategorized. Therefore, we are going to have to design our own pathway. So due to the COVID-19 pandemic, we are unable to gain access to laboratories, meaning that the experimental part of our project this year will be focused on dry lab techniques and modeling. We created this framework to guide us through our computational techniques, which will ultimately result in a dry lab-based proof of concept. We use a multi-step retrosynthetic approach to design the pathway from our desired compound, hypohydronic acid, to a precursor molecule in the E. coli metabolome. To do this, we first use RetroPath. The proposed pathways, and they're then compiled and validated by RP2Paths, the next tool that we are using. And then the enzyme selection is done with cell enzyme to identify the best enzymes to carry out our pathway. These enzymes will then be cloned into plasmids. We will document this process for the wet lab team next year to guide their experimental protocols. We are also expanding our dry lab to include constraint-based modeling. We are currently developing a flux balance analysis of our design pathway. Overall, the purpose of dry lab is to demonstrate we can produce our compound in E. coli by designing a novel pathway. This will act as our proof of concept. And so this is an example of RetroPath and RP2Path's output. The first and second steps of our framework. On the center of the screen, you can see the RetroPath's UI. This software performs the retrosynthesis. It basically compares our compound, hypohydronic acid, to the compound found inside the E. coli metabolome. And what it does is from there find a precursor in this core metabolome or core metabolism of E. coli. Then we feed the output of RetroPath to RP2Path, and this second tool organizes the reactions and precursors to form the pathways you see here. We use simple spontaneous organic reactions to begin our pathway, and then RetroPath identifies the enzymes catalyzed reaction needed to complete it.
with St Andrews on our human practices due to the similarity of our products. We are conducting various stakeholder interviews with dermatologists, sunscreen companies, potential consumers, and coral reef researchers to their concerns and values. We are also performing survey analysis, finding out people's sunscreen habits and their reactions to different synthetic biology solutions. This will inform us how to market our project in a commercial way. We're also collaborating with our host today on a meta-analysis. Due to the pandemic, we've had a lot of trouble contacting stakeholders. In response to this, we are performing a thematic analysis of YouTube videos talking about. This allows us to reach a global cohort of stakeholders and see real people's responses in the content of videos. From this analysis, we've been able to identify huge consumer and in-depth understanding of different ethnicities' perception of sunscreen and kind of gauge a prediction of how the world might respond to our project. In particular, we identified a generational shift in the presentation of sunscreen, beauty, and a health product. Another major find is to see videos produced by and those targeting people of color compared to fair-skinned individuals. It was clear from our analysis that issues facing people of color, such as hyperpigmentation and white cast, are not discussed as greatly as issues facing fair-skinned people. And finally, we've encountered some criticism about the limited success of our project. We understand climate change, and therefore coral bleaching is always going to be a problem. While any reduction in oxidative stress will reduce the coral bleaching, it will not solve the issue completely. So to ensure that we take a positive effect on our world, we're using our project as an opportunity to educate people about climate change and draw the issue to the problem. From completing our human practice work, we have finalized our project values. The project must be affordable. Most consumers recommend under £10. They must be suitable for individuals, including a wide range of shade options that leaves no white cast. We must be willing to expand our views beyond Western ideals and take in concerns of different groups of individuals. Our information must be well structured to prevent consumer confusion. We must be transparent with our people and the impact of climate change through the story of our project.
So this year, due to the removal of a wet lab element to our project, we have decided to explore new learning avenues and produce a complete business plan for Hyposol, the hypothetical company behind this new sunscreen. Our business plan includes several commercial analysis and characterization of the sunscreen market. On this endeavor, we quickly encountered new language and skills we had not been exposed to as biologists. This whirlwind learning experience was a large adaptation, so we decided to produce an essential entrepreneurship learning resource to help other iGEM teams to get comfortable with this new content. The purpose of our resource is to teach iGEM teams basic entrepreneurship skills, to help them determine if they could begin a startup and begin the process of creating a career from their iGEM project. This resource is publicly available now, and if you complete it, you get a ribbon that can be put on your wiki, demonstrating that you have engaged with this content and considered this side of the project. We are also mentoring our partner team, St. Andrews, and held a webinar at the Global iGEM Meetup, talking about the benefits of entrepreneurship and teaching some basic skills. So thank you everyone for listening. If anyone has any questions, you can contact us through any of these platforms on the screen.
Thank you very much for that interesting talk. By now, some of you might think, okay, iGEM is great and the teams are doing really good research, but why do they do a science slam and put such an effort into organizing it? Well, the short answer is we simply enjoy science slams a lot. Like, each of our team members watched dozens of science slams and we still enjoy it every time. I think it's just a great possibility and way to learn something new, and it doesn't even feel like you're actively learning something. The long answer is a little bit deeper. Besides simply having fun at this event, we also want to spread the word about science, and of course also especially about synthetic biology. We think that science was in the past, is, and always will be highly important for the society. There are so many examples for that. You just need to watch the news to see that. Think of Corona. Now the world is listening to scientists, and they really care about what they research and about the results. So we think that more and more scientists should start communicating their research. Moreover, I think that we should also take people's fears from laboratories, because most people only see a lab on pictures or even in science fiction movies. So I'm not wondering that they might have completely wrong associations with the lab and that they might feel intimidated or even frightened when they see these huge lab doors with all the dangerous symbols. So I think we as scientists should start and show the world that we don't sit in this ivory tower of scientific research, and also show that we step out into the world and show what we're doing into this lab. And this is exactly what our iGEM teams are doing. They have amazing projects, and I'm happy that we still have 3 amazing science lamps to hear. So let's come back to this image of a huge laboratory door with the danger symbols. One thing that happens in real life in a lab are repetitive tasks that cost time and effort. Nowadays, there are possibilities to make that easier for scientists, and one thing is the automation of such processes. The iGEM team from the Imperial College in London is working on such processes. So I'm really curious to hear how their project is going on and what they have to say about that. Welcome to the stage.
Hi, who are we? We are SoapLab, a team of 14 Imperial College students from different backgrounds that are taking part in the iGEM competition this year. Our project's aim is to make lab automation accessible for small-scale labs. Let's first consider why accessible automation is still not that common in labs. We learned that manual labor for repetitive tasks is time-consuming, and it reduces the reproducibility of scientific experiments. The true potential of automation is not yet apparent, as wet lab scientists are only beginning to want more scalability and streamlining of their experiment. In addition, as coding skills are required to write and debug complex workflows, intuitive and user-friendly tools. But what is exactly lab automation? Nowadays, total laboratory automation is a mix of numerous technologies, such as robotics, computers, and liquid handling. For more than a century, automation has been motivated by the same reasons: to save time and to improve performances through the elimination of human error. In the past, as is often the case today, the growth of certain industries or the demands of specific analytical challenges became the focal point for technological efforts. It is often the end user or bench scientist who has seen the need for automation being the driving force for the creation of an amazing array of devices. Let's go back in time and analyze the roots of nowadays technology. The first mention of any automated device in the American chemical literature was in 1875 by Thaddeus Stevens, professor at the University of Indiana. His device was designed to drip water through a piece of filter paper at a controlled rate to wash a filtrate. Progress in the field of automation continued in the early 1900s, with automated carbon dioxide measurements introduced in power plants. During the ensuing decades, there was an overlap of process control automation and laboratory automation. Real-time scientific control of production processes opened the door for automation in the laboratory. In 1929, an automated treatator was introduced, using a photocell to sense the color change to an indicator at a treataton's endpoint. During the presentation at the New York Electrical Society, the inventors claimed that the device was 165 times more sensitive to color differences than the human eye. By the end of the Second World War, the use of automatic control equipment in chemical manufacturing had become routine, and there was a new need for trained instrumentation experts. In response to this demand, the Chemical Engineering Department at Washington University incorporated experiments where students learned the operating characteristics of temperature control equipment. Starting in 1948, computers used in automation appeared in literature. The first computer mentioned was the analog computer, allowing for the first time chemical researchers and automatic process control equipment designers to create electronic simulations of their processes. A digital computer for laboratory instrumentation surfaced in literature in 1952. The Atlantic Refine Company announced that it had developed a mass spectrometer and digital computer combination capable of performing complex molecular analysis. The first truly automated instrument was introduced in 1957. The instrument, the Autoanalyzer 1, used continuous workflows analysis to increase the instrument throughput, 20 samples at a time, to leap past the manual process of the time. This one instrument started to automate the clinical laboratory workflow at the level of the assay. During the 50s, the one electronic technology that truly promised to revolutionize the laboratory was the transistor. When used in conjunction with a system of fast-responding sensors, it would allow for the collection of thousands of data points. In 1959, the first microchip is patented at Kilby. The other development that excited the imagination of scientists was the information appliance of the 70s. Electronic data management was brought to the laboratory to manage the workflow and electronic interface to the instruments, removing paper-based logs and capturing data electronically, directly from laboratory instruments. During the 80s, the evolution of motor and microprocessors technology enabled the development of the first true automatic liquid handling workstations. The current ones have all evolved from these early basic designs, adding laboratory unit operations functionality and software sophistication. OpenTrust began as an iGEM project in 2014, where the team worked on an open source equipment development in the form of a liquid handling robot. The project grew to a startup with $10 million funding round in 2018. Let's look at the impact of automation now. We consider the effect of implementing total laboratory automation in the Department of Clinical Laboratory at the School of Medicine in Shanghai. As TLA has been implemented, the data shows an increase in immunology test volume by 89% and an increase of chemistry test volume by 77% in 3 years. Moreover, the data shows a sharp increase in workload, almost double that in 2016. These results highlight significant workflow and financial benefits with the implementation of a TLA system. So let's state the advantages that automation is bringing. Introducing automation in labs improves in experimental reproducibility and scalability due to the standardized approach that is implemented and may reduce systematic errors. Moreover, automation facilitates higher production rates and a more efficient use of materials. Although high initial capital is required, in the long term, automation has an economical advantage and allows for more complex workflows. Automation leads to shorter work weeks in lab, from approximately 70 hours in 1900 to about 40 hours nowadays. Protecting lab workers, it reduces the biological risk that operators have to face. However, automation has its own downside. Automation brings in a question of reliability, since it leads to less human control on the experimental flow. In contrast with manual processes, automation has higher costs in the short term, higher maintenance, risking downtime and system failures. Another downside affects lab workers. As experimental techniques become easier, unqualified staff, such as undergraduates, can take part in research, sometimes working overtime. Consequently, staff trained in specific technologies are often fired, leaving operators unready to solve emergency situations. Automation can lead to dependence when a researcher relies completely on it, becoming constrained by machine potential limitations and losing manual skills. Once an experimental workflow is automated, limitation of a machine potential must be taken into account when considering further research developments beyond the current setup. Let us have a look on how automation and synthetic biology are working together. Optimization of strains and pathways is often a crucial step towards getting record product yield or striking the right balance of component activity in a genetic circuit. The power to be able to search within the biological productivity space requires high capital to afford automation. Current design tools also often lack integration with synthetic biology standards or have a good way of capturing variations of a construct to test lots of different combinations. OpenTrust and other affordable and open source automation systems promises less human error, enhanced reproducibility and cost reduction due to the optimization of material usage. We can conclude that lab automation is a very powerful tool, allowing for optimization of workflows through DOE and reproducibility of experiments. However, there is a learning curve to using them, introducing a new problem to overcome. Hence, we want automation to be accessible, easy to learn and exciting. For instance, during this summer, we have developed a software tool, SOPLab, to promote liquid handlers use. The researcher only runs the protocol created by our software on their OpenTrust to carry out the experiment. Our method allows for the benefits of automation without sacrificing accessibility. If you want to learn more about the ethics of automation, scan the QR code and join us for the webinar on the 16th of October. Thanks for being with us.
Thank you very much for that, Slam. Now, I want to tell you a small story from my personal life. During summer vacations, I give chemistry courses to children in the age of 10 to 12 years. One experiment I conduct with them is the synthesis of plastic out of starch. When introducing this experiment, I usually talk with the children about the places where to find plastic and also what to do with used plastic. And I have to admit, I was positively surprised how much they already knew about this problem and how worried they were. The iGEM team from the Ashesi University of Ghana is also aware of the problem of plastic pollution. And now I am really curious to hear how they combine the problem of plastic pollution with synthetic biology. So, Tamisha, the stage is yours.
Thank you. So hi everyone, my name is Tamisha and I'm the student leader for Team Ashesi Ghana. Before we start, I would like to thank Ashesi University for this opportunity. I'd also like to thank the iGEM team Heidelberg for even considering us to be a part of this slam. And I'd like to thank Revive and Restore for giving us a grant to be able to participate in the competition this year. And finally, I would like to thank my amazing team because they've been so great, me a lot of support during this time. So welcome to our Science Slam. Another day, another home being destroyed, another family being displaced by the menace that is coastal erosion. And this is just one of the villages in Ghana and in Africa. Another day of work, wading through this filth. Another day of placing his life on the line so that the volume of plastic can reduce. And this is just one landfill site in Ghana. What about the others? What if I told you that over the past couple of years, the there's been a drastic increase in the amount of plastic waste generated in Ghana. That's right, over 1 million tons are generated in Ghana each year, and the majority of that escapes into the ocean. As if that wasn't enough, the menace of coastal erosion slowly destroys coastal villages, like Fuveme mentioned earlier. In the past 12 years, 42% of houses have been destroyed, with an overwhelming 37% of the land being covered by the sea. This amounts to over 80 different families being displaced. When will this stop? It only takes one more bottle, one more wave. But we are here to bring a ray of hope into a seemingly dark situation. We are the Coastbusters, and this is how we plan on changing the narrative. This is our bioconcrete tetrapod, home to our trainee superhero bacteria. Let's dive in and meet them. Meet V. plasticina, one of our engineered bacteria and our first trainee superhero. She's been injected with a super serum that allows her to biodegrade shredded plastic, more specifically PET. Once she comes into contact with plastic, she uses PETase, a plastic degradation enzyme, to break down the PET into another component, MHET. Thankfully, once she comes into contact with MHET, she uses yet another enzyme called MHETase to break this down into two main parts, terephthalic acid, known as TPA, and ethylene glycol, known as EG. Ethylene glycol is a sticky substance, think of jelly or glue, that helps V. plasticina's colleague, V. sementin, to do what he does best. This is V. sementin, another engineered bacterium and our second trainee. His super serum contains genes from urolytic bacteria in order to help him precipitate CaCO3 particles. What happens when he is deployed is that he uses urease to hydrolyze urea in order to generate carbonate ions. These carbonate ions then bind with calcium ions to form calcium carbonate precipitate, and you can think of this as chalk or a white powdery substance. The amazing part of CaCO3 is that it helps to keep the sand particles together, and this is where ethylene glycol also comes in. Due to its sticky nature, it is much easier to hold the sand together and to keep the shape of their home. Our hope is to line these tetrapods along the coast of coastal villages to serve as a natural sea defense system against the crashing waves. But what happens if there's been a breach or a crack is made in the home? Well, a crack means that light has been introduced into a rather dark environment. Thus, the bacteria's superhome has a mechanism that allows it to repair itself once it senses light within it. This is made possible once more due to the biocementation work of V. sementin. He will have their home patched up and sealed in no time. Take a look at the image on the slide. Isn't this beautiful? Isn't this lovely? Well, we thought so too. Thus, meet the last member of the team, Lumen. Lumen is unlike her counterparts in that she's A completely different bacterium, and B, she shines in the nighttime, as you see in the picture. Lumen is a bioluminescent bacterium, and her presence in their home means that anyone who sees the tetrapod is amazed by how beautiful it looks at night. Now, I don't know how many of us have lived with roommates before, but I can say that sometimes you don't always agree. And that's the case with V. plasticina and V. sementin. When plasticina uses her powers, the environment becomes increasingly acidic due to the presence of the terephthalic acid. However, when sementin uses his powers, the environment becomes increasingly basic. Due to their differences, we will make sure that their home is self-calibrating. Once plasticina has had time to work, the acidic environment will enable one of our pH-inducible promoters, think of a sensor, to wake Simonson up and tell him to work. As Simonson works, he increases the pH and makes it more basic, which then enables another pH-inducible promoter to wake Plasticina up and get to work. Think of our pH-inducible promoters as alarm clocks, and think of our bacteria as taking shifts. This is how our system will regulate its internal pH to make sure that our superhero bacteria are in sync. We're still in the modeling phase of our project and haven't gotten a chance to build a scaled-down version of our bioconcrete tetrapods. However, we believe that this is a way to help curb two problems at once. The plastic will be collected from plastic landfill sites, washed and shredded to be used in the tetrapods. And the tetrapods themselves will be lined along the coast to prevent the waves from washing away more villages. And that's our team of superhero bacteria. I hope you've enjoyed learning a little bit about them and our project. Once again, my name is Tamisha, representing Team Ashesi Ghana, and thank you so much for listening. Have a good day.
So thank you, Tamisha, for that slam. I'm a little bit sad to announce that this was already the last science slam from our guests. But this doesn't mean that we're finished yet. Definitely not. Now it's time for you at home to vote for your favorite slam. So how does this work? We prepared a poll in which you can decide which slam you like best. So the link for that will be pinned under this live stream, and we also prepared an image with a code which you can scan. So please vote. Moreover, I didn't say yet what the winner team will receive. Well, we prepared a couple of really nice stickers, and they will also get some magnets for their fridge or whatever lab fridge. And also in 2 minutes, our own team will present their research. So Mario is already preparing for his slam, which you can hear soon. But now it's time for you to vote. So,
Angela, this might be the right time to talk about our experience this year with Corona, for example. I'm quite sure it was a hard year for all of you, and so it was for us. Angela, how was it for you to work with iGEM this year with these complicated situations?
well, you're definitely right. Well, it was quite challenging. Well, I came to the team a little bit later than the others, and what I experienced was a great atmosphere, even though we couldn't meet in presence and physically, but we had great team meetings held online, and definitely. And yeah, well, we were struggling a lot with getting lab access. This was really difficult at the beginning.
Yeah.
But well, our dry lab team is working really hard and they make progress every day. So I'm really happy to hear that.
I'm also happy. And as I hear, the vote is ready. Is it right? Yes. So now it's your time to give your voice for the best science slam of our guests today. And I hope you will enjoy my presentation coming up in a few minutes. Thank you very much for your votes, fellow iGEMers. iGEM is a contest of bioengineering, but what does that mean, bioengineering? When I think of an engineer, who first comes to my mind is the great Leonardo da Vinci. He was not also a clever scientist and painter. He analyzed nature and designed complex, functional machines. And this is very much like iGEM, where we analyze nature up to its molecular detail and design molecular machinery such as fusion proteins. But there is, of course, more in engineering than just analysis and design. Unlike Da Vinci, the modern engineers have two core concepts, foundations of engineering, which are modularity and standardization. Two core features also very known to iGEM. For example, for our biobricks. For those unfamiliar with iGEM, the biobricks are short parts of DNA, which are shared with the community, modular and highly interchangeable. And this is a concept of modern engineering, making a whole crowd of people able to engineer and design things, making a universal genius like Da Vinci obsolete. Well, we'll figure out if we need him anyways. Further, a machine needs to be constructed to prove its design, to prove its functionality. And sadly, most of Leonardo da Vinci's designs were never been built. Some have been too expensive, but most of them were simply huge and far ahead of the construction methods of its time. You know that, fellow iGEMers? What about you? Didn't we all have ideas of proteins and molecular machinery far too big for a single cell of yeast? This is very much a problem in bioengineering. What we are missing are molecular assembly line are standardized parts like screws and threads perfectly fitting into each other to switch and interchange modules. And this is also a core concept of the modern wonder of engineering, the International Space Station. It wasn't assembled on Earth and shipped to space. No, engineers divided the design and conquered the Earth's orbit just to be able to have this wonder flying around our beautiful planet every day. So, what is our project? What's the idea? We're going to give bioengineers the tools to assemble structures far beyond the possibilities of a single plasmid, such as small proteins. And therefore, we want to link them together and assemble them with RNA, which you can see here, are parts of our cells, the transcripts of DNA, design blueprints that make up every cell of us. But RNA is not only this transcript, it's also argued to be the origin of life, and it shows very interesting structural properties, such as loops, which can be recognized by proteins and even alter their behavior in some cases. And RNA is highly specific in its binding due to its individual sequence. The RNA is highly recognizable for proteins. And we want to establish a standard for these binding sites to be able to assemble broader machineries in a cell after expression. However, standardization is not the best way to go, especially for real-world problems. Because proteins are not just these simple, goofy figures flying around the screen. No, proteins are far more complex. Because what you see here is the three-dimensional structure, and what we make in the lab is often just the sequence which needs to be folded in place, a sequence of amino acids. perfectly shaping the protein. And if we want to bind RNA, for example, with this protein, only a few of these amino acids luckily bind to the RNA. So therefore, mutations in these regions binding the RNA are more crucial than mutations in other regions. Nevertheless, finding the right structure to bind a specific RNA can be pretty challenging. And there's no help with standardized parts if you want to come up with something completely new. So I guess Da Vinci is not obsolete yet. We need these genius minds. But who of your team could it be? I mean, who would be that genius? For our team, we developed an artificial intelligence, machine learning algorithms, which have the knowledge and experience of hundreds of years of studying, of scientists exploring the structures of proteins. In our case, we fed our algorithm with 500,000 known RNA-binding protein sequences, each having up to 50,000 individual features, such as the charge of a protein or its structural properties. And this algorithm, and you can see it here, where the protein is fed in, this algorithm is not only knowing these sequences, it also can generate new sequences. Given some features and given its experience, we can generate new feature-finding RNA binding sites, completely new protein RNA. Well, not completely new, it's analyzed by nature. But let's take an even closer look at this, because it's nice to have this structure on paper, but as I said, the folding is something pretty important about proteins. And therefore, we want to predict the folding before we go to the lab. And so we need another part of machine learning, this time a neural network fed with the generated structure, and it shall predict a three-dimensional structure to show us if it has these features we wanted to have and we selected for in the process before. This back transition gives us the possibility to validate the sequence before we go to the lab. And that's where I want to finally take you. What are we doing in the lab this year? As we don't have that much time as in other years, we want to prove our concept at first. There is something called split GFP. It's a fluorescent protein which is split into two parts. Each at once, not functional at all. But if you bring them together, it can emit light if you give it some light of a different wavelength. So what we want to try to prove our concept of linking proteins with RNA is to take our RNA linker with the designed protein domains and thereby fuse these proteins together, making the GFP glow again and signaling us that we have got a match of our proteins. This is, of course, not all you can do with this mechanism. Think, for example, of broader structures. Even this is small compared to what we can see and observe in cells. The interesting part about the core concept, standardization, modularization of these linkers, is that you can simply switch some of your molecules pretty fast. And even more, you could build up whole libraries of fusion proteins with modular complexes fused together. And this is something we really know from nature. Our antibodies consist of exactly such libraries, built up with other mechanisms, but the functionality of our immune defense is highly dependent on modular design. And these days, why not to look at our immune system? I want to thank you all for listening. And at last, the great engineer Da Vinci might not be obsolete. I thank you very much for listening. Thank you very much for your contribution. And of course, our fine moderation, Angela, and the technic team behind the camera. Thank you all for making this possible, and I'm giving the final word back to Angela. Very thank you much.
Thank you, Mario. I'm a little bit sad and surprised how time is running. Can you believe?
Pretty fast. We didn't expect time running that fast.
Definitely. So we didn't arrive at the very end yet. First of all, I want to say thank you to the Studierendenwerk. Thank you for the possibility to have this cozy facility. Well, we really missed it to be here and drink coffee.
It's a beautiful place. When you ever stay in Heidelberg, go to the Studierendenwerk, the Marstall Café. It's beautiful here, yeah?
Exactly. Moreover, we want to thank our sponsors. Such a huge project like our iGEM project wouldn't be possible without your help and your support. So thank you very much. We really appreciate it. Moreover, I want to thank you as spectators who are still watching the slam. We're really happy that we have so many spectators today. And I also want to thank you as iGEM team who participated today. I know it's a lot of effort and time that you spent doing these slams, but I think it was definitely worth it. I enjoyed all of them. So thank you very much. And now it's time to announce the winner team.
Finally.
I'm really curious. I received this envelope and I don't know what's inside. So we don't know the winner yet. Do you want to?
Yes, I can hold this for you.
So, I'm really curious. Our winning team for today's Science Slam is the Ashesi University from Ghana. Tamisha, congratulations. Well, you will receive your package with some stickers and a winning magnet and maybe some other things too in the next couple of days. Thank you very much for all the other teams. You will also receive post from us. Not as big, but you will also receive something small. Now, I want to say goodbye, good evening, and I hope that we could share our enthusiasm for synthetic biology. I hope you will watch many more science slams. And now, good evening.